Hello everyone, so this is the second part of the video of lipid metabolism. In the first video, we had already talked about the detailed process of beta oxidation and how it happens in 4 steps in an even carbon fatty acid chain. This is the second part of the same video where we are going to talk about oxidation of odd carbon chain fatty acids, oxidation of unsaturated fatty acids, and what is peroxisomes and how beta oxidation is related to that. We'll also talk about Zellner syndrome and synthesis of ketone bodies. That is an important part of beta oxidation in lipid metabolism. So let's start. As you can see in this slide, so mitochondrial beta oxidation is highly efficient process, but it requires some standardized procedures. For example, mitochondrial beta oxidation requires a straight, even carbon saturated chain. But if the fatty acid doesn't have a straight, even carbon or saturated fatty acid, what if it has an odd number of carbons and it is unsaturated? What will happen then? How will be oxidized? So in those cases, the product cannot enter TCA cycle, but requires specialized enzymatic adapters to reshape and fuel for the TCA cycles. So let's talk about those specialized enzymes in this video. So 3 carbon leftover problem. Right, so we already talked about in the previous video. If you hadn't watched that, go back to my channel and watch the lipid metabolism part 1 video. We are talking about where we have talked about palmitoic acid, that is 16 carbon fatty acid degradation. So what happens in this, we have talked about in the previous video. It is a 16 carbon. In every cycle of beta oxidation, 2 carbon will be removed as acetyl CoA. This is a 4-step process. 4 steps will occur in each cycle, and at the end 1 acetyl CoA will be removed. So after each cycle, first it will be 16, then there will be 14 carbons left, then only 12 carbons left, then only 10, then 8, then 6, then 4, and the 4 will be divided into 2 carbons in the last cycle. So 7 cycles will be required for palmitoic acid to be degraded. But what if there is this is an odd number of carbon? Instead of 16, it is 15. What will happen then? So as you can see, the first steps will be doing the same procedures, but instead of 14, there will be 13 number of carbons left. After 2 carbons are removed, 13 left, then 11, then 9, then 7, then 5, then 3. So at the end, in an odd number of carbon fatty acid chain, 3 carbons are left at the end. So we are going to talk about how to solve this problem. So 3 carbon leftover problem. The oxidation of a saturated odd chain fatty acid proceeds normally until the final round, and instead of yielding 2 carbon units, it leaves behind 1 acetyl CoA and 1 3 carbon fragment. Right, so this 3 carbon fragment propionyl CoA, this 3 carbon compound is called propionyl CoA and is an oversized puzzle piece, requires dedicated metabolic detour. Since this 3 carbon compound cannot directly enter the TCA cycle, it requires 2 carbon acetyl CoA to enter the TCA cycle. So since this is a 3 carbon, this cannot directly enter the TCA cycle or citric acid cycle. So what will happen? This is the propionyl CoA rescue pathway, the 3 carbon CoA rescue pathway. The 3 dedicated enzyme reshape the 3 carbon fragment into succinyl coenzyme A. As you know, succinyl coenzyme A is a molecule that is required for TCA cycle or citric acid cycle. This is an important molecule that could enter the TCA cycle. So what will happen is there will be 3 enzymes that will convert this propionyl CoA into succinyl CoA. succinyl CoA so that it can enter the citric acid cycle or TCA cycle. So how will that happen? So here is propionyl CoA. It will use an ATP and carbon dioxide and undergo carbosylation. It will undergo carbosylation. What is carbosylation? So carbosylation is addition of one carbon. So this is either through addition through a carbon dioxide or carbonate molecule. So this propionyl CoA, which is 3 carbon molecule, adds another carbon to it and produces a 4 carbon dimethylmanonyl CoA. This procedure requires an ATP and a carbon dioxide. This carbon dioxide oxygen is added to this, so that's 3 carbon is changed to 4 carbon, and this process is carboxylation. The enzyme utilized in this is propionyl CoA carboxylase enzyme. The enzyme name is propionyl CoA carboxylase enzyme, and the cofactor required for this enzyme is biotin. So at the end, the 4 carbon dimethylmanonyl CoA is produced. Now this dimethylmanonyl needs to be converted to L-methylmanonyl CoA for it to undergo the final step. D is converted to L through an enzyme called methylmanonyl CoA epimerase. Once this epimerase converts it into L-manonyl CoA, the next step is L-methylmalonyl CoA is converted to succinyl CoA, which can directly enter the TCA cycle. And this happens by the enzyme methylmanonyl CoA mutase. This mutase enzyme helps in this production of this molecule. And the cofactor required by this mutase is vitamin B12. Now here is a problem. Few people get a deficiency of vitamin B12. In case of deficiency of vitamin B12, when body cannot produce or there is no vitamin B12 in the body, what will happen? The final step of the rescue pathway relies entirely on vitamin B12. If there is no vitamin B12, this mutase enzyme, this methylmanonyl CoA mutase enzyme cannot act. And if it cannot work, mutase enzyme, then L-methylmalonyl CoA cannot produce succinyl CoA. This procedure is broken in case of missing vitamin B12. So, a B12 deficiency paralyzes this enzyme, causing a toxic accumulation of methylmanonyl CoA that degrades the peripheral nerve function. So, in case of vitamin B12 deficiency, L-methylmanonyl cannot be converted to succinyl CoA and this cannot enter the TCA cycle. In that case, L-methylmanonyl CoA is accumulated in larger and larger quantities that goes nowhere and is accumulated inside the body. This is toxic. And this causes a condition called peripheral neuropathy. Thus causes nerve degradation due to its accumulation. Now moving on to the next slide, the unsaturated problem. Now we had already solved the problem where there is odd chain of carbon, but what if the carbon chain is unsaturated? So saturated chains are generally straight, while in case of unsaturated, where there is double bonds, kinks are produced. These kind of bends and kinks are produced in cases of unsaturated fatty acid. Now a saturated straight molecule of acyl CoA can form a perfect fit within the acyl CoA dehydrogenase enzyme. In case you have forgotten, I will remind you, as we have talked about in the previous video, that acyl CoA dehydrogenase is the first enzyme for oxidation required for the oxidation of acyl CoA. This is the first enzyme that is required, and it utilizes FAD to produce FADH2 and produces trans delta 2 enoyl CoA. This is the first step of beta oxidation, this enzyme. So in case of a saturated fatty acid that has no double bonds, the molecule is straight and can easily fit inside this enzyme activation site, active site. While in case of unsaturated cis bond chain, such kinks are produced, such bends are produced that cannot enter inside this acyl CoA dehydrogenase enzyme active site. The enzyme slot is blocked. There is a steric blockade due to 30-degree bends. Now moving on to the next slide. What happens? So isomerase and reductase straightening technique is utilized here. To resolve the double bond blockers, the cell deploys isomerase to shift bond position cis to trans and reductase to resolve the complex polyunsaturated intermediates. So cis 3 enoyl coenzyme A is produced that has a 30-degree bend. An isomerase enzyme straightens this and produces trans 2 enoyl CoA. This is now a straightened trans configuration. And as you can already tell, the even numbered polyunsaturated bonds already produces this molecule, right? Like we already talked about, in the second step of beta oxidation, this molecule is already produced. The unsaturated acyl CoA in an even numbered unsaturated acyl CoA undergoes acyl CoA dehydrogenase and produces this trans 2 enoyl CoA. In case of even numbered polyunsaturated bonds, unsaturated acyl CoA produces 2,4-dienoyl CoA that undergoes a reduction procedure and produces cis 3 enoyl CoA, and again this isomerase converts it into trans 2 enoyl CoA, which is already produced in the second step of beta oxidation. This will undergo hydroxylation. Moving on to the next slide. Very long chain, the spatial anomaly. Mitochondria cannot import very long chain fatty acid VCLF C20 to C26. These oversized lipids must first be routed to peroxisomes for initial heavy duty trimming before the shortened remnants can enter the standard processing. So a molecule that has over 16 carbons cannot easily enter the mitochondrial matrix, right? For example, there is VLCFA carbon 24. It cannot enter the mitochondrial matrix. So first it have to be enter inside the peroxisome enzyme for pre-processing and cut into little pieces before it could be imported into mitochondria for beta oxidation, right? Now the peroxisomal furnace, heat over ATP. Unlike mitochondria, peroxisomal oxidation hands electrons directly to oxygen, creating highly reactive hydrogen peroxide. The enzyme catalase neutralizes this threat, releasing trapped energy entirely as heat. So FADH2 produces H2O2, and catalase enzyme, this is a toxic molecule that is produced during the cutting off of fatty acid into little shortened pieces, and this produces H2O2, hydrogen peroxide, which is toxic for the body. So this peroxisome has an enzyme called catalase that removes the electron and produces H2O molecules and releases the rest of the energy as heat. No ATP is synthesized here. Now Zellweger syndrome. Let's see what that is. A rare genetic disorder characterized by the complete absence of functional peroxisomes in all tissues. Cerebrohepatorenal syndrome, without any initial oxidation, very long chain C20 to C26 fatty acid are accumulated toxically. Since there are no peroxisomes, there are no organelle that could cut up long fatty acid chain into short fatty acid chain that could be sent inside the mitochondria. Since there are no peroxisome to cut off long chain of fatty acid, these long chain fatty acids of C20 to C26 are accumulated toxically inside the body and causes Zellger syndrome. This is due to lack of peroxisome, and it causes cerebrohepatorenal syndrome. Niche processor alpha and omega oxidation. So when standard beta oxidation is blocked or overwhelmed, specialized tissue deploy alternative degradation mechanisms. Here you can see alpha oxidation occurs in brain tissue and omega oxidation is a minor pathway that happens in endoplasmic reticulum. This alpha oxidation removes one carbon at each time. COA, this must be COA. One carbon in each cycle, unlike beta oxidation that removes two carbon. This removes only one carbon at a time and there is no coenzyme A involved. While in omega oxidation, this is a minor pathway that happens in endoplasmic reticulum. Cytochrome P450, O2 and NADPH is required for this procedure to occur. Now oxidation matrix. Let's see the difference between a diagnostic comparison of fatty acid degradation pathway across cellular locations. So there is mitochondrial beta oxidation happens in mitochondrial matrix. It requires even straight chains. Energy is high ATP yield and there is no clinical pathology involved. And there is peroxisomal beta oxidation that oxidizes large molecules like C20 to C26 fatty acids before it can enter into the mitochondria. It happens inside peroxisome. No ATP is produced, only heat. And in case the peroxisome is missing, it causes Zellger syndrome. In case of alpha oxidation, it happens in brain tissue. Branched fatty acids are produced. Energy yield is less than beta oxidation and removes one carbon at a time. At each cycle, only one carbon is removed. Then there is omega oxidation. It happens in endoplasmic reticulum. Standard fatty acids can undergo this and doesn't require NADPH, uses cytochrome P450 instead. Now systemic overflow, the ketogenesis trigger. Ketogenic bodies are another things that are produced during beta oxidation. When the liver beta oxidized liver faster than the citric acid cycle can process the resulting acetyl-CoA, it activates an overflow pathway condensing the excess into transportable ketone bodies. So as you know, after beta oxidation, acetyl-CoAs are produced and this acetyl-CoA enters TCA cycle or citric acid cycle. But if beta oxidation is happening at a very high rate and this acetyl-CoA is produced in very large amounts, while all those cannot enter TCA cycle because this is happening slowly. So this excess acetyl-CoA that remains inside the body is transformed to produce ketone bodies, right? So you can see in this procedure triggered by starvation or untreated diabetes, there is a flood of acetyl-CoA is being produced and citric acid cycle's capacity is over. No more citric acid cycle can take acetyl-CoA. So the overflowing acetyl-CoA undergoes this process and produces ketogenic bodies or ketone bodies. This happens when there is diabetes, if the patient has diabetes or the patient is fasting, a person is fasting. This is a ketone synthesis tree. HMG-CoA serves as the central processing node, cleaving acetoacetate. From there, the pathway branches yielding stable metabolic fuel or spontaneously decaying into volatile acetone. So, acetyl-CoA is produced after beta oxidation. It forms a condensation reaction with another acetyl CoA to produce acetoacetyl CoA. This acetoacetyl CoA then produces HMG CoA, 3 hydroxy 3 methyl CoA, and thus acetoacetate is produced. This is broken down into D3 hydroxybutyrate or acetone. This acetone can be smelled. This is a specific smell that you can actually smell in patients of diabetes, the acetone smell. Right, so this is the ketone pathway in diabetic patients. Now, survival versus toxicity. During prolonged starvation, the brain shifts to ketones for survival. However, in untreated diabetes runaway, overproduction leads to toxic blood accumulation and ketoacidosis. D3 hydroxybutyrate and acetoacetates are two things that are produced under ketogenesis pathway. When starvation adaptation starts, there is no glucose. So acetone is sweet smelling breath. Untreated diabetes also show ketoacidosis. The metabolic fate map. Every alternative pathway ultimately converges on just two molecular keys, acetyl CoA or succinyl CoA, ensuring the central biological engine never stops running. So there is odd chain fatty acids which undergoes to produce propionyl CoA that will produce succinyl CoA and enter the citric acid cycle. Then there is peroxisomal, there is also will be even chained, it is not mentioned here. They will produce acetyl CoA like this and enter inside the citric acid cycle. It could be unsaturated fatty acids or peroxisomal remnants which will again produce acetyl CoA and enter into TCA cycle. And once it enters into TCA cycle, if the acetyl CoA cannot enter the TCA cycle, it will undergo ketogenesis. Acetoacetate will be produced through 3-dehydroxybutyrate and produce ketone bodies. So this was the final fate map. Thank you for listening to me and watching this video. If you like the content, please like, share and subscribe the video.